Good afternoon. I would like to welcome all of you who have dialed into our investor call, in which we will review the recent research note we published, Genetic Medicine's 2021 Year in Review and Expectations for 2022. My name is Keay Nakae. I'm a senior healthcare analyst here at Chardan. Joining me on this call is my colleague and fellow senior healthcare analyst here at Chardan, Dr. Geulah Livshits. As call participants are aware, our focus at Chardan is on identifying companies that can generate exceptionally high long-term investment returns by creating shared value for society through disruptive innovation. Before we get started, just one compliance issue. During this call, we will be discussing our published note that contains all of the relevant compliance disclosures. With that said, for anybody in our audience who'd like to submit a question, please type it into the chat box. So let's get started. 2021 again saw significant scientific platform development and clinical validation advancements by genetic medicines focused companies, which we believe should continue into 2022. Despite that, 2021 proved to be a challenging year for biotech stocks, and particularly those in the genetic medicine sector. After hitting an all-time high in August of 2021, the NBI went on a downward slide to finish flat for the year, underperforming the S&P 500 by 25%. The downward momentum has continued into 2022, with the NBI declining another 14% to date and the XBI down 20%. While this performance reflects serious valuation concerns for the sector as a whole, we believe it has created a more attractive entry point for selective biotech investors. We believe that the fundamentals remain strong, with continued discovery, development, and clinical validation apace, with large pharma cash positions supportive of M&A and collaborations, positions the sector for a stronger performance in 2022. So with that, let's get started. And let me hand it over to Geulah to discuss gene therapy and gene editing.
Thanks, Keay. So as Keay said, 2021 was a tough year for segments of genetic medicines along a few different dimensions. Safety was a big topic of discussion on systemic AAV in vivo gene therapy. Bluebird ran into issues commercializing its cell therapy in Europe. There's a number of open questions relating to allogeneic cell therapies, though over the year, bright spots have been Intellia's in vivo editing proof of concept and the COVID vaccines. But even their valuations have drifted back from peak, putting us broadly in the space, as Keay said, at a potential solid entry point. So let me elaborate on those points a bit. So specifically for vector gene therapy, as I mentioned, 2021 was a rough year as there were ongoing questions about systemic AAV safety, as well as to some extent a dearth of important catalysts that could reinvigorate some interest in the space. In 2022, the field is adapting to the cautious FDA in terms of safety. This week, we did see a Logic Bio get a clinical hold after a serious adverse event of thrombotic microangiopathy in a clinical trial. That's not gene therapy, gene editing, but also uses AAV technology. But thanks to a greater experience with these type of safety events, we're seeing companies adopt increased immune monitoring and are catching these adverse events earlier, opening some avenues for mitigation that could still enable attractive risk-benefit profiles. And based on the FDA's panel on AAV gene therapy from last September, it doesn't sound like there's going to be a one-size-fits-all strategy, but companies are applying what has been learned, for example, incorporating sirolimus or complement inhibition, or potentially CD20 depletion in immune suppressive regimens, but in a way that takes disease and overall risk-benefit into account. We've seen parallels to some extent in the CAR T space, where the management of serious side effects like cytokine release syndrome have improved considerably over clinical and subsequent commercial experience and development. So obviously beyond that, new capsids could enable activity at lower doses or have less propensity towards some of these immune engagement side effects that we've seen and are of interest and are being actively explored in the earlier stages of research. That's another exciting avenue that we're watching closely. But in the nearer term, we do see potential for programs that are using some of these earlier generation AAVs like AAV9, like Rocket's Danon program, to achieve a viable risk-benefit profile for commercial use. That ties into another theme that we've talked about in the research. Rocket's updated data on Danon disease that's expected later this year could have positive read across to other AAV9 heart gene therapy programs from Tenaya and Renovacor, which are expected to enter the clinic this year as well. So generally, the in vivo gene therapy space has been dragged down by a number of factors, but we should also keep in mind that there are regulatory catalysts on the horizon in the space that could shift the discussion to a more commercial conversation. These include BLA or MAA filings from Crystal and Unicure CSL on epidermolysis bullosa and hemophilia B, BioMarin's Valrox submission for Hem A, and potential details on a regulatory path for Keay's giant axonal neuropathy program, either filing directly or comparability studies. So within the next 18 months, there is potential for the list of approved gene therapies to look quite different than it currently does. And I should add that locally delivered gene therapies have been a bit more insulated from the safety issues that we've seen. The challenges here have been more about interpreting readouts, whether biomarkers or the complex multiple readouts that are associated with ocular gene therapies. But there have been some encouraging data sets like Mira's radiation-induced xerostomia study, and we expect additional data on that front in that program this year. And ocular gene therapy readouts from Mira and Janssen, as well as AGTC in the first half of this year, could reinvigorate momentum for that space as well. For CNS gene therapies, data from companies like Passage and Taysha could help establish some of the links between the biomarkers that we've seen and potential relation to clinical benefits as that space continues to mature with either intrathecal or ICM delivery. And then I'll just touch on gene regulation and logic gates. We've been talking about synthetic biology for a couple of years now as an exciting area of science to watch in biotech, and now we're getting actual programs that are getting ready to make the jump into a clinical development. So the space is maturing. It's going from science project to having product candidates in place or nearing the nearing further clinical development. And so this includes Mira GTX. We wrote about their RiboSwitch platform in December, and they plan to file an IND this year, first just testing the small molecule inducer itself, and then eventually in combination with a gene regulatory cassette. But what's exciting about this type of platform is that it's in principle modality agnostic. It can be used in the setting of an AAV vector, some other vector, or an engineered cell therapy and control expression of various transgenes. So that's the type of technology with broad applications that we could see driving things like value-adding partnerships for Mira and other companies.
Let me ask you a question. I mean, what's an example of where that type of technology could be useful here?
Yeah, so internally, Mira plans to use this with local delivery, so salivary gland and ocular encoding peptides or hormones or antibodies. But I've also said this type of technology could be interesting for regulating something like CRISPR or enzyme expression in tissue settings, for example, where AAV is still your best bet for mode of delivery, but you don't necessarily want to have your gene editing enzyme active long-term due to potential cumulative risks of unintended editing outcomes or something like that. So, but there's a broad area in terms of the types of cargoes that could be delivered where you want tight regulation of expression and will want to be able to turn something off after you've initially had it on for some amount of time. But actually, in terms of gene editing, maybe let's switch to that. In 2021, gene editing, as I mentioned, had the landmark first in vivo editing data from Intellia in transthyretin amyloidosis, where we saw deep reductions in the target TTR protein after lipid nanoparticle delivery of the CRISPR components to the liver. And that was a major proof of concept for the space and led to, I believe, 117% performance for the year. And then other gene editing companies that were using similar LNP in vivo strategies for the liver also ended up at the top of the gene editing performance basket that we track. Although those were obviously in the preclinical stages still. So overall, a good year for LNP. But in 2022, we'll see maturing data from Intellia's lead program, as well as its second program in hereditary angioedema, which basically uses the same platform, just with a different guide RNA and target sequence. So we think it has a good shot of showing positive editing data, which could drive additional performance there. So open questions for those programs are more along how the in vivo editing translates to potential clinical phenotype modulation and comparisons to things like RNA-based approaches and other modalities that are either available or in development. And then more broadly on the in vivo front, we'll see entry of base editing into the clinic from Verve, which we don't cover, but also use an Lnp delivery platform. And in 2022, we expect to have an update on Editas's ocular editing program in LCA10. Editas has kind of lagged behind its cohort of public CRISPR peers that have been in the clinic, as we haven't yet seen the clinical proof of concept definitively. But here the readout is more complicated because there's not a validated circulating biomarker or a clear readout that is easily interpretable, and there's multiple different ocular metrics. So we could see some improvement on maturing data and data at higher doses and from that update. But in terms of Editas, we also think that Editas could get a boost if they do a partnership on their iPS cell-derived NK cell platform, which we've written about a number of times, and I'll get back to deals in a moment. But on the ex vivo side, more generally, we'll see some regulatory progress in 2022 as Vertex and CRISPR have guided to filing on their sickle cell and beta thalassemia therapy CTX001 by the end of the year. And also on the regulatory front, I should mention that the FDA's CBER have on their agenda to put out draft guidance documents for the development of gene therapy products containing genome editing. So this could provide some clarity on expectations around analysis of things like unintended genome editing consequences or things like that of various editing approaches that are in the clinic and also in development. And we could see some moves towards standardization of these types of assessments. But from a bigger picture standpoint, one of the topics that we've written about in the past years has been our expectations regarding deal flow between gene editing companies and developers of complementary technologies. And this certainly played out in 2021 and shows no sign of stopping in 2022. And a lot of this is tied to the expansion of the gene editing toolbox, base editors, now prime editors, various targeted insertion strategies, and other gene writing approaches, as well as the characterization and engineering of novel CRISPR enzymes by various private and public companies in the space that we've mentioned in our research. And we expect that to continue with demand for access rising in concert with the toolbox expansion. And we think ultimately most credible cell therapy platforms will eventually want to have access to some type of editing platform. And we're now also seeing more mature editing companies internalize technologies, as we saw earlier in Beam's Guide acquisition and Intellia's acquisition of Rewrite Therapeutics that was announced this week.
So Geulah, let me ask you a question about all of this development. You know, how do these emerging next-gen technologies, you mentioned base editing, prime editing, fit together? Do these newer ones supplant, let's call it 1.0, which is a double-stranded break, or you know, how do all these, both the first wave and then the second wave of companies, you know, pivot to utilize everything in the toolbox that you just mentioned?
Mm-hmm. Yeah, so I don't currently view it as one technology superseding or supplanting all others or another in all cases. I think there's not one tool for every application. In some settings, for example, high efficiency is more important. In others, it's extremely important to have a high degree of specificity, and you want to be really confident in the type of editing outcome that you're getting. In certain clinical indications, there's multiple different mutations that, let's say, drive a disease indication, in which case a gene replacement strategy might be more commercially viable than having to develop 30 different products that correct each of the individual mutations. But those are all things that can evolve as the space matures. And then in other settings, for example, where AAV is the only route of delivery, things like size of the system and the cargo is a major determinant of what would be feasible. So for now, based on where the field is, matching the right tool for the job is important. And so that's where we're focusing our evaluation. But in general, as I mentioned, delivery, I think that's been a really important theme in gene therapy and gene editing, and I suspect in the RNA space as well. So Keay, why don't I hand it over to you to talk about RNA?
Great, thank you, Geulah. So in 2021, RNA Therapeutics maintained their strong momentum, driven by continued clinical, regulatory, and commercial success. particularly in the vaccine space. Obviously, the rapid development of the effective mRNA vaccines to address COVID-19 was a once-in-a-generation event that affirmed the capabilities of mRNA technology and jump-started the development of new mRNA applications, which importantly now will be leveraged by the accelerated investment in the manufacturing build-out. With respect to small RNA therapeutic targets, expansion into tissues outside the liver continues to progress, facilitated by new conjugated targeting moieties, again, addressing delivery, as you just mentioned, Geulah. In terms of performance, the publicly traded RNA companies that we track in our portfolio showed a cap-weighted return of 139%. Again, similar to 2020, the COVID-19 pandemic vaccine development efforts really was driving this outperformance of the mRNA subgroup. So with respect to mRNA, I think that the question is, really what's the next act? I think the first thing we're going to see is the companies like Pfizer and Moderna, who are already developing mutation or variant-specific new payloads. And again, this is one of the attractive features of mRNA, the ability to quickly match the payload to the target. So we should see throughout 2022 seeing data from those efforts. Not all of the mRNA companies did well. CureVac shares lost about 58% of their value. Arcturus was kind of up and down, eventually ending the year down 24%. But we did see one acquisition, and that was Sanofi's acquisition of Translate for $3.2 billion. Again, as we move forward, I think we'll see more efforts by the mRNA companies, specifically in the vaccine space, to develop some new products. This can be Moderna's flu vaccine, CMV vaccine, and an Ebola vaccine. And then Sanofi also has a flu vaccine. So I think, at least for mRNA, the vaccine applications we'll see continue to be the lower hanging fruit, if you will. The therapeutic efforts are maybe further back. For RNAi, we continue to think this sector is poised for continued growth. At the end of the year, we saw the FDA approve Novartis's Leqvio. It had been approved a year earlier in Europe. Finally got approved in the FDA, in the U.S. by the FDA, and this really represents the first large market application for an RNAi. Alnylam positioned to receive significant royalties up to 20% from Leqvio sales by Novartis. Alnylam is obviously the leader in the RNAi space with 3 commercial products. They did generate a total revenue of $662 million, which represented 83% year-over-year growth. The 3 products being Onpattro, Givlaari, and Oxlumo. Up next for Alnylam, I think top of mind here is what will happen as they seek to get a label extension for ATTR from the current polyneuropathy to cardiomyopathy. We already know that the next generation version of Onpattro, which is vutrisiran, is looking very good. The company has already reported positive results from the Helios-A study, most recently 18-month follow-up data. It does have a PDUFA date of April 14th, but again, that's for the polyneuropathy label. I think what everybody is really anticipating is how well these drugs will do in the cardiomyopathy population. And first up will be Apollo B data. That's again for Onpattro, and we should see that in the phase 3 for cardiomyopathy patients in the middle of 2022.
Okay, for the RNAi-focused companies, where else would you expect them to be most successful going forward?
Yeah, I think for RNAi, what's clearly de-risked here is the ability to deliver an siRNA sequenced to target an mRNA and be able to knock it down or silence it, specifically when delivered to the liver. And again, I think there's ample evidence that they're able to deliver safe and potent doses of their therapeutics to the liver, facilitated primarily by the ability to use GalNAc delivery, which is very specific to the ASPG receptor that's expressed on the liver hepatocyte cells. So any initiative where they're targeting the liver and they're targeting something that has a validated target, whether it's a gain of function or loss of function mutation, at this point, I think there's a high probability that they're going to be successful. I think one example of that is both Alnylam and Arrowhead are both advancing RNAi candidates targeting a specific gene, HSD17B13, for the treatment of NASH. This loss of function mutation is associated with lower odds of developing NASH. And I think notably Arrowhead, which only presented its first interim results from its phase 1/2 at EASL in May of 2021, when we rolled into November of 2021, they were able to announce an exclusive licensing agreement with GSK for this asset, everything outside of China, which included $120 million upfront. So again, just based on some interim phase 1 data at this point. Arrowhead also has liver-targeted programs targeting ApoC3 and ANGPTL3, which they envision will be large addressable markets. Even within that, the company just started a phase 3 for ApoC3, targeting adults with the genetic mutation FSC. Now, again, this is a smaller subset of patients, but again, where there's a clear validated target, and so where we would have high expectations of success. All the RNAi companies currently have an asset to address chronic hepatitis B. All of these are in phase 2, evaluating a combination therapy approach. Again, the goal of the RNAi component of the combo therapy is to knock down surface antigen, and we're seeing kind of consistently across the board, whether it's the Arrowhead compound that's partnered with Janssen, Alnylam's compound that they're working with Vir on, or the Dicerna compound where they're working with Roche. We're seeing, kind of consistent, very good knockdown of surface antigen, and we're seeing the same with Arbutus with their AB729. That remains the only one that's unpartnered at this time. Another large disease area is Alnylam. Its RNAi, zilebesiran, targeting hypertension. It's now been advanced into 2 phase 2 clinical studies as both monotherapy and add-on therapy in patients with uncontrolled hypertension. So again, I think that could be a good market. Again, a liver target and a validated target. So I think to answer your question, that's where RNAi has really been de-risked. As you mentioned before, delivery continues to be a key enabling component for small RNA to move beyond the liver. We continue to see more programs moving beyond the liver. Arrowhead itself has a program, HIF2, for renal cell carcinoma. We'll see some additional data within this quarter on that program. They also have a program for their first muscle target, which is DUX4. Alnylam, their first exhepatic RNAi is ALN-APP, targeting amyloid precursor protein for the treatment of Alzheimer's disease and cerebral amyloid angiopathy. In their case, they're using a different conjugate. It's a lipophilic conjugate, which they refer to as C16. They believe this can be utilized to deliver its siRNAs to CNS, eye, and lung cell targets. Although we don't cover either of these 2 companies, Dyne and Avidity both are developing oligonucleotide therapeutics using a different conjugate. It's an antibody as the targeting moiety. So using the navigational properties of either a full monoclonal antibody in the case of avidity or the antibody fragment in the case of dyne to selectively target a cell receptor. Both of their initial programs are targeting the transferrin receptor on muscle cells. Avidity is already in the clinic, dosing the first patients with its lead program, AOC 1001, targeting DM1. They expect to advance additional candidates in 2022 for DMD and FSHD. Dyne has submitted its IND for Dyne 251 for DMD, and they also have programs targeting DM1 and FSHD as well.
Okay, we're seeing a lot of the newer RNA companies like Dyne, Avidity, and Chardan focused on muscle diseases, going after some of the same targets or indications like DM1 and DMD. So what distinguishes one program versus the other in terms of trying to predict which could be more successful?
For DMD, I think it's really simply about trying to get more of the drug to the target than what Sarepta using a naked PMO construct has been able to do. So both Dyne, Avidity, and Chardan, which again we don't cover, are all using a PMO construct, but again with their own conjugate to deliver more of the payload to the target. So I think there's less differentiation for DMD other than the conjugate. For DM1, there is quite a bit of difference between the different programs. In the case of Avidity, they're delivering an siRNA, so they're trying to utilize RNAi interference by engaging a risk complex that exists in the nucleus. In the case of Dyne, they're delivering an ASO, so they're trying to utilize the properties of that ASO to attack or break up the pre-mRNA that's binding the protein. In the case of Entrada, they're delivering a PMO. So PMOs are neutral, very stable, and in their case, it simply displaces the ability of that protein to get bound up. I think three different approaches, three different conjugates, and I think for DM1 we're going to see, perhaps different results from the three companies. Moving on to LNP delivery, again, we talked about the fact that we have the successful COVID-9 mRNA vaccines developed by Moderna and Pfizer-BioNTech. I think this clearly reestablished LNP as a very valuable, safe, and robust delivery vehicle. That said, those particular applications do utilize intramuscular delivery, so it really does represent local administration. I think system administration of LNP really is currently limited to the liver, and this is due to the affinity of apolipoprotein E or ApoE, that once the LNP with the cargo is administered IV, the PEG layer kind of comes off, and then the ApoE proteins get absorbed onto the neutral liposomes in circulation, and then there's a subsequent preferential uptake of ApoE by low-density lipoprotein receptors, or LDLR, which are expressed on the surface of liver hepatocytes. So nevertheless, even though it is, at this point, LNP delivery is really limited to just the liver, as you talked about, Geulah, Intellia's landmark readout utilized LNP. for their CRISPR mechanism for HATTR. Verve has reported using LNP to deliver their mRNA gene editing to target PCSK9. So I think one of the development initiatives we'll look to see going forward is how to be able to utilize LNP to get it to targets outside of the liver. I know there was a paper that we referenced in our note that described the sort approach used by Recode, in which in addition to the traditional 4-component lipids of the LNP, they add a 5th component, and the idea is to deabsorb the ApoE to make the construct have less of a preferential uptake by the liver, and by adding some other elements to it that perhaps make it more amenable to be taken up by the spleen or the lung. So that's certainly one approach that's out there being developed to, again, enable LNP to deliver cargoes, whether they be mRNA or gene editing outside of the liver. The one final area I wanted to talk about was an increase in RNA-based editing, specifically ADAR-based editing. We saw a number of both collaborations and financings here during the year. ProQR entered into a collaboration with Eli Lilly for their ADAR-based editing approach. We saw a collaboration between Shape Therapeutics, which is private, and Roche. We saw ADARx be able to raise $75 million in a Series B. And then WaveLife, which we don't cover, also has an ADAR program. They've presented some very interesting early data for their program targeting alpha-1 antitrypsin deficiency. And we'll look to see more of that development in 2022 on the ADAR front. So with that, let me hand it back over to you, Geulah, to talk about cell therapy.
Great. So just on the topic of going beyond the liver, within cell therapy, we're seeing cell therapy mature, but there's still a number of logistical and biological challenges within the ex vivo cell therapy space. So we've also seen some increased momentum in parallel for targeting cells of the blood or immune system, cells that are often used within cell therapy applications. But we're seeing momentum for targeting those cells in vivo. And we ask if that's ultimately years or decades down the line, but could be the future of cell therapy. And in short, we're not there yet, but there's a lot of work in this area by academic groups, biotech companies, as well as signals from big pharma that this is an area to watch. The companies that have been operating in this space include Intellia, Moderna, and Beam, which have each reported delivery to hematopoietic stem and or progenitor cells in preclinical models, as well as Emergis and Sana and several others and academic groups that have are working to develop or establish in vivo CAR T programs. Delivery is obviously key here, and the extent of the specificity and distribution patterns would likely guide the kinds of applications that will establish the low-hanging fruit in terms of risk-benefit with these types of modalities. But in the nearer term, we're still seeing the ex vivo cell therapy space developing rapidly due to investments in the science, in the manufacturing, and in the logistics and symptom management. We saw approvals for some autologous CAR T programs in 2021 in CD19 cancers and BCMA cancers, and there was just a publication in Nature from Carl June's group on some early CAR T patients that are now in 10-year remissions with some long-term CAR T persistence. So the cell therapy paradigm clearly has potential for long-term meaningful value, but there's also a lot of open questions. So on the allogeneic cell therapy front, in our 2021 preview, we said we'd want to see durability data to understand how allogeneic cells compare to autologous CAR Ts, which are increasingly moving into earlier treatment lines and in clinical evaluation, and bispecifics, which in theory have simpler logistics. And so far, to some extent, the question is still open as CRISPR therapeutics and Allogene are moving into consolidation dosing paradigms rather than the one-time paradigm that was established for autologous CAR Ts. But there'll be more data readouts this year. So this year in 2022, we could see additional data and have a better understanding of what durability looks like. And similarly, we expect longer-term follow-up data from different platforms, including NK cells, gamma delta T cells, to compare the activity and durability as well as safety across these different modalities. But again, there's a lot of open questions on both the biology and the technology front, and that includes which, for example, in the gene-edited CAR T or CAR NK space, there's questions around which targets to modify to get cells that sufficiently expand in a patient's body for enough time to get the right area under the curve to actually clear a tumor, so that encompasses multiple parameters: ability to expand, not to be exhausted, not to be cleared by various immune system cells, and then different allo CAR T programs in the space are all using slightly different strategies. And then NK cell programs also use distinct cell sources like iPS cells or donor-derived, et cetera. So there's a number of different variables that as the field matures and we see data on more patients with longer follow-up, we'll start to see some resolution and daylight between the different technologies. And meanwhile, this year, we can also see some increasing momentum for TCR, T-cell therapies for indications outside B-cell cancers. Intellia has a WT1 targeted program that's in the clinic for AML. Another TCR-based AML program is an HA1 targeted program from T-Scan Therapeutics, which also has a cleared IND. And for solid tumors, Immatics has multiple solid tumor programs in the clinic. And the more important readout on patients treated with a likely go-forward dose would be in 2022. They had some initial data in 2021, which we thought were encouraging, but we think the higher dose has potential for meaningful durability. So we expect to be hearing more about TCR therapies this year, and the oncology field in general has been talking about cell therapies for solid tumors for years, and it's been slow going, but the field can also get bolstered to some extent by momentum if Iovance's TIL therapy clears its potency assay hurdles and moves through BLA filing. Obviously, that's a different technology, not TCR, not genetically engineered, but signals that that cell therapies for solid tumors can achieve meaningful outcomes could bolster the space more generally. And similarly, on the TIL front, we expect to get some data from Achilles on their platform that focuses on clonal neoantigen reactive TILs. So overall, in 2022, a number of data readouts across the cell therapy space that could answer some longstanding open questions and give a better sense of how the different modalities fit in with each other. And so keeping mindful of the time, I think we can get to some questions. We have time for a few questions. And as a reminder, if you'd like to ask a question, please enter it into the chat box. We've already gotten some come in, so let me go through that. So we have a question on which individual names you have conviction on today that have catalysts this year. So with respect to probability of success and any other parameters. So I think that's for both of us. Keay, why don't you go ahead?
Sure. I think we would stick with our two leading companies in RNAi, Alnylam and Arrowhead. Both have sold off quite a bit here. And another company's data, BridgeBio, for their ATTR program failed to hit the primary endpoint. In that case, the placebo group did better than expected. And there's been a lot of Monday morning quarterback since then to try to determine whether or how accurately those patients were assessed. Nevertheless, we believe that for Alnylam, as I said earlier, expanding the label to include cardiomyopathy patients is a big opportunity for them. We will or expect to see top-line data from the phase 3 Apollo B of patisiran in cardiomyopathy patients. What we've seen up to now that would give us confidence that we could see a good result in that patient population is that in both Apollo and Helios A, so for the phase 3s for patisiran and vutrisiran, respectively, in the polyneuropathy patient populations, where there were subsets of patients with cardiomyopathy, all of the assessments of those patients, I think, are all lining up in the right direction. So I think a positive readout from Apollo B would be a significant catalyst for Alnylam. For Arrowhead, it's just kind of more a collection of all the things that they have in the clinic. And we will see, as I said, a readout or more data, I should say, on HIF2 this quarter. I think getting further clarity for their AAT program will be helpful as well. We'll see additional data from their Sequoia study, 12-month biopsy data. And I think with that data in hand, it'll further clarify the regulatory strategy to take that program forward that's partnered with Takeda. And then for both of those companies, as I mentioned, we would expect to see additional data throughout the year for their hepatitis B programs, and then just additional interim data for their other various programs sprinkled throughout the year. So for us, those would be the two names.
Got it. So for me, I can answer that as well. I think I mentioned 3 for the moment. That would be Rocket, Mira, and Intellia. So Rocket, as I mentioned, presented some data on its Danon program. That's an AAV9 gene therapy for genetic heart disease in 2021. I thought the data were encouraging. The market had a somewhat negative reaction. And I think there's potential to see some rebounding in value, especially with a greater visibility on the regulatory path and updated data. And that's more of a second half of the year catalyst, but it could also serve as a rising tide, lift all boats scenario catalyst for the earlier stage cardiac gene therapy programs from Tenaya and Renovacor. And then Mira, they had an encouraging readout in radiation-induced xerostomia late last year, and interesting data. And as I said, more on that this year. And plus, we expect to have data from the XLRP program with Janssen. And Janssen featured this collaboration last year in their investor day. They didn't have to do that. It suggests that the collaboration is going well to us. And again, we think the riboswitch platform is an interesting and differentiated technology, and you could see additional visibility on that could be interesting. But the XLRP data, we think, could be a positive catalyst there. And Intellia, they're steadily amassing a nice collection of genome editing platforms for different applications, as traditional CRISPR editing, some homology-independent targeted insertion type of approaches, and now gene writing. There's interesting collaborations outside of the core areas. And as I just said, in terms of probability of success, I think the second in vivo editing program has a good shot of reading out positively in terms of target editing. I think we have another question regarding themes that we may have picked up over the past few months that could dictate things like which modalities garner more attention. Okay, you want to take a shot at that one?
Yeah, I think really it's the moves outside the liver for small RNA. Again, we've kind of identified some of the conjugates that are allowing, at least initially, the move into muscle. We talked about Alnylam's use of C16 for their applications in CSF, the lung, and the eye. And there's a lot of opportunities out there to move beyond the liver. There's a lot of opportunities within the liver, as we also talked about. But getting outside the liver, the programs are earlier. The questions are still unanswered to some extent. But I think we'll see, again, with those initial programs from Dyne and Avidity, as they report out data, we'll see, if positive, increased confidence in both those approaches and then the opportunities that they can open up to other targets outside of the liver. I know in the past, Avidity has talked about having maybe a dozen other antibody receptor targets that they feel could work.
Yeah, I think my answer is pretty similar to yours in terms of focus on delivery and collaboration around that. Whether it's looking at next-gen lipid nanoparticles or next-gen AAVs, or understanding the parameters that make a particular vector suitable for a particular indication. This is a theme that's been of major discussion and a theme where we've seen collaborations cropping up in the past several months. So I see we are pretty over time or at time. So I think we can wrap up here. And if anyone has additional questions, we'd be happy to make time to follow up individually if you reach out to your Chardan sales representative. And thank you everyone for spending the time, and have a great weekend.